Hey guys, today we're going to be talking about what are Punnett squares, which are kind of a visual tool, more of a visual diagram, that is quite commonly used throughout genetics. It is mainly used in the field of determining the different genotypes of an offspring when two different individuals are crossed. So that also brings up the topic of how are genes crossed in general. When two genotypes are put together, how does an offspring form its own genotype from those two original genotypes? So in this video, we're going to answer these two questions and see how these two explanations come together to form a very interesting phenomenon within life science. So let's go ahead and start off with the definition of Punnett squares, which is right here, a visual method of calculating the possible genotypes in the offspring of a cross between two individuals. So let's go ahead and break down this definition. A cross between two individuals. So when two individuals are crossed, the offspring is going to be developing, and as it's developing, it's receiving thousands and thousands of genes, thousands and thousands of traits from its two parents. So how does this offspring develop its own genotype from these two parents? So I'm going to go ahead and explain it through an example. So let's say our two genotypes for a certain trait is heterozygous, are both heterozygous. So both parents for this random trait, it could be anything, are heterozygous. So what the offspring is going to do is, in order to form its own genotype, is it's going to take one allele from one of the parents and one allele from another one of the parents, the other parent actually, and it's going to put those two alleles together to form its own genotype. So with that being said, let's go ahead and look at the math, or more the probability of this. So for the first allele, it has two different options, right? From this genotype, it could either take a capital G or a lowercase g. And then for the second allele, it again has two different options. It can take a capital G or a lowercase g from that second genotype, from that second parent. So there you have two different possibilities for two different alleles within the same genotype. So knowing some probability, in order to find out the total different possibilities of genotypes, you'll multiply 2 times 2, two possible alleles paired up with two other possible alleles, and you get 4, meaning that you have 4 different possible genotypes that the offspring could possibly have from when two individuals are crossed. So a Punnett square is merely a visual method of representing this cross. It's a visual method of showing the 4 different possible genotypes, the 4 different crosses that could result from the two individuals. So now let's go ahead and see how a Punnett square is drawn, how this visual method works. So here, I have drawn a square with 4 quadrants, and each of the quadrants is meant to represent each and every one of the 4 genotypes that the offspring could possibly get. So again, I'm going to use the same example that we used before, the 2 heterozygous individuals for that certain trait, and we're going to go ahead and cross these 2 individuals. So how it works is we normally take the first genotype and assign it to the top side of the Punnett square. And we're going to put each allele to each of the halves of that specific side. So we're going to put the capital G on one half of the top and the lowercase g on the other half of the top. So as we're putting each of the genotypes to one side, it is very important that you put the same genotype on the same side. Like you shouldn't mix up the 2 genotypes. Like you couldn't put a capital G and a capital G on the top. That would mean that you're crossing a purebred, not a heterozygous, if that made any sense. So, again, we're going to put the second genotype to the second side of the Punnett square, and generally we use the leftmost side of the Punnett square. So we only use the top and the leftmost side of the square for the 2 genotypes. So, again, we're going to put the capital G and the lowercase g at one of the halves of the square. So now, as we're pairing, as we go throughout the square, I should say, we're just going to be pairing the 2 different alleles on its side. So we're going to go to the top for this first quadrant, we get a capital G, and then we're going to go to the side, and we get another capital G. So that's how it works. So you go to the top and to the left, and you get those two different alleles, and you're going to pair them together. So let's do that again for the second quadrant. So on the top, there's a lowercase g. And on the side, we're going to go all the way to the side, and you get that capital G. So in our genotype, we always put the capitals first. So it's going to be a capital G, lowercase g. Now let's move on to this quadrant, and we're going to go to the third quadrant, and we're going to go to the top again, and we go all the way to the top, and we're going to get the capital G over here. And when we go all the way to the side, we get the lowercase g. So as we're pairing this, let's see how it plays out in the actual genotype. So over here, this top G is the first capital G within that first genotype, right? So this capital G is the first possible allele from the first genotype. And so when you come down here, the lowercase g is the second possible allele from the second genotype, and the capital G on this side is the first possible allele from the second genotype, and the lowercase g on the top is actually the second the second allele from the first genotype. So hopefully that wasn't too confusing, but that kind of gives you how that all played out in the Punnett square. So again, now let's go to the fourth quadrant of the Punnett square, and over here, we're going to go all the way to the top, and we have this lowercase g, and when we go all the way to the side, we also have a lowercase g. So now we have a completely recessive. So over here, you can see that when we cross two heterozygous individuals for a certain trait, we have one chance of a complete dominant, a complete purebred for dominant. We have one chance of a complete recessive for dominant. And we have two chances for a heterozygous. So as you can see, the Punnett square gives us a very visual method of pretty much just doing the pairing that I kind of showed you as I was doing all those red circles after we paired the third quadrant. So from this Punnett square, you get that visual method of, you get kind of a statistic of the possible identity, of the possible genotype of a certain offspring. Like we have a 25% chance of, or a 1/4 chance of a complete purebred for dominant. We have another 25% chance for a complete recessive, and we have a 50% chance of a heterozygous. So hopefully that was helpful, and thank you for watching.